Good morning, everyone. Welcome to Session 6, Top Abstracts. I'm Arpita Desai from the University of California, San Francisco, and I'd like to introduce my fellow moderator, Dr. Abhishek Tripathi.
Good afternoon, everybody. This is Abhishek from University of Oklahoma.
Thank you, Abhishek. We'll start off with a few trivia questions. How many drug-drug combination therapies have been approved by the FDA in the last 15 years? So it looks like the trivia question 2 was pushed. We'll move on to the trivia question 1. How many drug-drug combinations have been approved by the FDA in the last 15 years? And the answer is C. I'll let my fellow moderator, Dr. Tripathi, move on to the next session.
Great. Welcome, everybody. We'll start off this session with our first presentation, which will be from Dr. Eric Jonash. He's a professor in the Department of Genitourinary Medical Oncology at the MD Anderson Cancer Center, and he will be providing us an overview of the funding mechanisms for RCC in the next few minutes. Dr. Jonash.
Thank you very much, and thanks to the organizers for this amazing meeting. Very cool platform, great content. And what I'm going to talk about in the next 10 minutes are the funding mechanisms for renal cell carcinoma. So renal cell carcinoma really has been consistently underfunded relative to other diseases. And here are slides that I've obtained from Brian Lewis, which look at, for example, the American Cancer Society 2018 grants by cancer type. The red arrow points to the number of kidney cancer grants, 13. And then you can see the breast cancer grants, there's over 160. NCI funding is pretty similar, where you see the dollars spent per renal cell carcinoma death is in around $2,900 range. And you look at other diseases like brain, thyroid, and breast cancer, it's over 10,000. So clearly already there's a discrepancy between the number of deaths and the amount of money that's being spent by the NCI. Similarly, with private funding initiatives, if you look at, for example, melanoma, the number of cases and deaths per year, and then the private funding per year, melanoma has over $10 million in private funding. And renal cell carcinoma, until the Kidney Cancer Association came into play recently with some excellent funding, also was quite underfunded from a private funding initiative perspective. So there's a real unmet need to fund research in renal cell carcinoma. Fortunately, there are expanding choices for renal cell carcinoma researchers, and there are a couple of excellent opportunities that I'm going to quickly summarize here. First and foremost, the DoD Kidney Cancer Research Program, the Kidney Cancer Association grants, the AACR grant, and the VHO Alliance grants. The Department of Defense Kidney Cancer Research Program, through individuals like Brian Lewis and other people who really have worked very, very hard to motivate the funding through congressionally mediated funding. The the KCRP now has $40 million earmarked for renal cell carcinoma research in fiscal year 2021, with the major areas of emphasis that I outline here. There are several types of funding opportunities that I'm going to go through in detail here, and there are these 7 mechanisms that are currently available. The first is this Clinical Trial Nurse Training Award. So obviously having an infrastructure for clinical research is critically important, and here a principal investigator can be an individual who is a nurse who has a RN all the way up to a doctoral degree, and will create a mechanism where research nurses are mentored to perform kidney cancer research. The next is the Postdoctoral and Clinical Fellowship Award. Here you have individuals who have 3 years or less of postdoctoral or mentored clinical research experience who can get funding of $195,000 in conjunction with a designated mentor to perform kidney cancer research. And again, this is over a 3-year period, an excellent opportunity for very young faculty. The Concept Award is a high-risk, high-yield award where you basically have an emphasis on innovation. It's $100,000 over 1 year. Preliminary data are not allowed, so it's the great idea that you get at 3 o'clock in the morning that you can hopefully translate into money to develop that to a larger grant. The Academy of Kidney Cancer Investigators that Brian Rainey is going to talk about a little bit more. These are individuals who are within 3 years of their postdoctoral research. This is a mentored early career investigator-based grant with $725,000 in funding over a 4-year period to complete some type of research program that can go, project that can go all the way from basic to clinical research. The Consortium Award for Clinical Trial Sites. So MD Anderson and with me as the lead run the Kidney Cancer Research Consortium. The idea here is that we're going to have a mechanism where we can perform mainly investigator-initiated clinical trials in a collaborative fashion, and we're creating the infrastructure to make that possible, and clinical trial sites can be funded to join this consortium. The Translational Research Partnership Award is an award with $750,000 over 3 years, where you have a partner between a clinician and a laboratory scientist to test hypotheses and move the field forward. And lastly, we have the Idea Development Award, where $600,000 in direct costs over 3 years, or depending on what other mechanisms there are, there's also the early detection and epidemiology options with more money, where funding for these usually mechanistically based projects can be obtained. The Kidney Cancer Association has also earmarked over $1 million for renal cell carcinoma research in fiscal year 2021, with 2 Advanced Discovery Awards and 4 Young Investigator Awards. The Advanced Discovery Award is again a partnership between a clinical investigator and a basic scientist to move forward hypothesis-based research into the next level of funding. And this can include biomarker development, development of novel therapeutic targets. So it's fairly broad. It's great, a great grant for a team of scientists. The Young Investigator Award is $75,000 in direct costs over 1 year period for fellow level individuals. And we're going to hear Pavlos Masul's Young Investigator Award immediately after this. The AACR-Exelixis Renal Cell Carcinoma Research Fellowship is again a fellow level grant, $120,000 over 2 years that can help develop a very young career investigator. And lastly, the VHL Alliance research grants are really focused mostly on VHL disease. Obviously, renal cell carcinoma is part of that. But if you have a grant that really looks at the fundamental mechanisms of VHL mutated diseases that can encompass both VHL disease and renal cell carcinoma, this is an excellent opportunity with $150,000 in the research project and $25,000 for the pilot project. So in summary, in the last 5 years, we've really had an increasingly large number of choices, funding choices for investigators in renal cell carcinoma. The other thing to bear in mind, as you've seen with this meeting, as well as other meetings, is we have an incredibly collaborative group of individuals that I really believe are second to none, and working together to help each other move the science forward. And that really adds value to every grant. I think the dollar figure of that it can't really be underestimated. And already we're seeing with this meeting, as well as other meetings, that we're seeing significant advances being made thanks to the support being offered. So thank you very much. And once again, congratulations to the organizers for this amazing meeting.
Thank you, Dr. Jonash. Moving on to our next presentation, we have the Kidney Cancer Association Young Investigator Awardee, Dr. Pavlos Masao, who is an assistant professor at the University of Texas MD Anderson Cancer Center. And he will be talking to us about targeting synthetic vulnerabilities in renal medullary carcinoma.
Thank you for the opportunity to present in this meeting. And on behalf of my collaborators, I'm very pleased to present our work on renal medullary carcinoma, which has been supported by the Kidney Cancer Association from the very beginning, including through the Young Investigator Award that I received in 2017. These are my disclosures. And focusing on renal medullary carcinoma, abbreviated as RMC, this disease was first described in 1995, and it predominantly afflicts individuals with sickle cell hemoglobinopathies, including sickle cell trait. And 1 in 14 African Americans will have the sickle cell trait. That's about 3 million people here in the US. And a good number of them, 1 in every 20,000, possibly even more frequent, we're finding out now, will develop RMC at a median age of 28 years old. And now we know that this is actually the 3rd most common kidney cancer in children and young adults. It is 3 times more common in men compared with women, and also 3 times more likely to happen on the right kidney compared with the left. The vast majority of patients will present with metastatic disease, and even if they don't initially, you wait for a few weeks or a few months and it will metastasize very fast. Untreated, it has a survival of 3 months at most. With our best available therapies in 2016 and 17 in a large multi-center analysis, the median survival was only 13 months. Clearly, we need to do better. RMC is always characterized by loss of the tumor suppression SMARCB1. Every case will have inactivation of SMARCB1, and this cancer is completely refractory to the anti-VEGF TKIs that we normally give for other kidney cancers. Response rates in the range of 0%. It does respond sometimes to platinum-based cytotoxic chemotherapy, but very often the responses are not durable. So clearly we need to do better, develop new ways to manage, diagnose this disease, and treat it informed by its molecular landscape. And that is what we did with the support of the Young Investigator Award. And a few related malignancies that I wanted to highlight here because they're very important in our comparisons that we show later. Colorectal duct carcinoma is a cancer that looks very much morphologically similar to RMC, but it is not characterized by SMARCB1 loss, and it is not associated with sickle cell hemoglobinopathies. Malignant rhabdoid tumors often happen in the kidney, and they are characterized by loss of SMARCB1. We abbreviate this cancer as MRT very often, but it doesn't look like RMC at all. Like under the microscope, it looks like an undifferentiated sarcomatous tumor. And finally, urothelial carcinoma, and particularly of the upper tract, is anatomically close to RMC, and it is also often treated with similar regimens as we treat RMC, platinum-based cytotoxic chemotherapy. So we published the results recently, a few months ago, in Cancer Cell of our comprehensive molecular characterization of RMC. We took untreated primary RMC tumors, and we were able to generate models with which we did mechanistic experiments, and we did a comprehensive molecular profiling that allowed us to identify certain hallmarks of RMC. One of it was the gain of chromosome 8q. The other one was monosomy of chromosome 22, where SMARCB1 is located. The other one, which was well established in a fantastic paper in European Urology by Calderaro and colleagues in 2016, that RMC is characterized by inactivating SMARCB1 translocations. But also we found that the RMC is also characterized by replication stress that is induced by upregulation of the MYC pathway, recurrent focal copy number alterations, as well as other hallmarks like NOX upregulation, UCA1, that's a link RNA upregulation, and upregulation of the C-gas-STING innate immune pathway. For this presentation, I'm going to focus on the replication stress and other stress-induced responses and how we can target this in clinical trials. That will be the focus of this presentation. So we were not surprised when we characterized RMC to find that it had a very low mutation load. When we looked at it by SNVs, you can see here highlighted in red, it's on the left side of this figure. Very low mutation load, similar in that sense to MRT. However, MRT, malignant rhabdoid tumors, have a simple genome overall, and we were surprised to find that RMC actually does not have a single genome otherwise, because it is actually characterized by focal copy number alterations, as shown here on these GISTIC figures with amplifications and deletions, and as well as at the arm level and whole chromosome level, we see a lot of 8q gains. And 8q is where the c-MYC oncogene is actually located. And fascinatingly enough, even if you see in a karyotype of this RMC cell line, you see that it's a mostly diploid cell line. But look how it has gained one extra copy of chromosome 8, and it even has 2 derivative chromosomes shown here that were derived from a translocation of 7q and 8q. So this cell line has at least 5 copies of 8q where c-MYC is located, which was very surprising to us. And then we looked at the transcriptomic profile, and we compare RMC with collecting duct. I told you why they look very similar under the microscope, and with upper tract urothelial carcinoma that is often treated with similar therapies. And we found that clearly RMC is distinct from urothelial carcinoma. It's a very different cancer, although it's treated very similarly currently. And it has a little bit of overlap with collecting duct, but it's still distinct. Interestingly, despite being both SMARCB1 negative tumors, MRT, which essentially looks more like a sarcoma, is very different from the carcinomas, as you see on the right side here. All of the carcinomas, including RMC, cluster separately, and RMC is clearly different in many ways than MRT. And one of the hallmarks that we identified, as I mentioned before, of RMC is this DNA replication stress. And you can see this at the transcriptomic level in these GSEA figures, where we compare RMC, primary untreated RMC tissues with adjacent normal kidney. And you see also that this replication stress is also high when you compare RMC with collecting duct carcinoma. Remember, the main difference between those two is the loss of SMARCB1. And indeed, when we took our cell line models, both 2 cell lines of RMC and 1 cell line of MRT and a cell line of SMARCB1 negative epithelial sarcoma, we basically did up assays where we restored SMARCB1, and we found downregulation of c-MYC, but also downregulation of pathways by Western blot associated with replication stress. When we did a down assay by knocking down c-MYC with siRNA, we found a very similar pattern. And to look more deeply into this mechanism of replication stress, we knocked down in HEC293 cell line SMARCB1, and we found an increased by DNA fiber assays shown here on the right side, increased replication fork speed when you knock down SMARCB1. This is an established general mechanism of replication stress and associated DNA damage repair response, as established by a paper by Maya Mendoza and colleagues in 2018 in Nature. And so we knew already that platinum-based chemotherapy, as well as other drugs like gemcitabine and adriamycin that can aggravate replication stress, actually can work, at least initially clinically, against RMC. And indeed, when we took our RMC cell lines, the two cell lines shown here, and we rescued SMARCB1, we brought back SMARCB1, which would attenuate replication stress, we found that the cells became less vulnerable to platinum salts and to doxorubicin and to gemcitabine, whereas the sensitivity to other chemotherapy drugs that have different mechanisms of actions like paclitaxel did not change. The same thing was found when we knocked down c-MYC. And so this points to a concept that was first established by our collaborator Gianni Genovese in Nature in 2017 in SMARCB1 negative pancreatic cancer, whereby the loss of SMARCB1 results in MYC-induced stress. And he first established it in, with regards to MYC-induced proteotoxic stress. And then Ali Carugo published a paper in Cancer Cell with Gianni and us showing that this proteotoxic stress can be a vulnerability for RMC tissues. And in our latest work, we're showing that this MYC-induced pathways also cause replication stress. And you can see here those pathways, proteotoxic stress and replication stress via MYC upregulation activated in RMC versus adjacent tissues. And with that in mind, with this mechanism summarized in this slide, whereby the SMARCB1 loss through the MYC upregulation, and the MYC upregulation is also facilitated by the characteristic gain of 8q, induces, the MYC upregulation induces replication stress that causes this sensitivity to topoisomerase inhibitors like doxorubicin or nucleoside analogs like gemcitabine or platinum salts. And at the same time, the c-MYC upregulation can cause proteotoxic stress that we can target by proteasome inhibitors. And that's the rationale behind our clinical trials shown here, which we activated in August 2018. And we've already enrolled 23 out of 30 patients projected, showing that indeed, even for such a rare cancer, we can enroll fast despite the COVID pandemic. And in this trial, we're testing the gemcitabine plus adriamycin backbone targeting replication stress, which is an established regimen for this cancer, with the addition of the proteasome inhibitor ixazomib to see if we can get better outcomes by targeting both MYC stress-induced pathways against this disease. I would really like to acknowledge the many people from all around the world that have supported this study and have worked on this study. I would like to acknowledge again the Kidney Cancer Association. This started from support by the Concord Cancer Foundation in collaboration with the Kidney Cancer Association that supported my first YIA, and then with the second YIA by the Kidney Cancer Association supporting this research, and this led to further support by the DoD, KCRP, RMC support sites, and of course I would like to acknowledge our patients. Happy to answer any questions. You can find me in these links.
Great, thank you so much for that great talk, Dr. Masro. Our next speaker is Dr. Brian Reaney, who's a professor of medicine at the Vanderbilt Cancer Center, and he'll be giving us a presentation on the Department of Defense mentorship program.
Thanks, it's good to be here. Eric mentioned to you the CDMRP family of grants, and the this mentorship academy was one of the mechanisms from about a year ago. And so I'm just going to briefly describe, what our vision, what my vision is for that. I was awarded to be Dean of the Academy, and then you'll hear from the 3 early career investigators who are now members of the Academy. So this is the vision for the Academy. So it's really about drawing young investigators into kidney cancer and keeping them there. with structured mentorship, providing them with opportunities so that they can become independently successful and, again, remain contributors to the mission of curing kidney cancer. So these are the components. I won't read it to you, but it's really about myself and an advisory panel who will have regular interaction with the mentees, really providing visibility and opportunities. That's key to mentorship, is efficiencies by the mentees, providing them with opportunities to succeed, focused training, regular feedback to the mentees. And we've kicked this off with an in-person, well, it wasn't an in-person workshop, it was a virtual workshop that was a couple days ago to go over projects, provide critical feedback. Even from that brief session, there was a number of collaborations and opportunities for interaction that were identified and we'll be moving forward with. To back up a little bit, this is taken from the grant, and when I was putting it together, I contacted about 20 or 25 early investigators and just asked them to tell me what were the barriers to academic success as they saw it. And you can see on the left here the different barriers and then some strategies, hopefully, that this academy can take to overcome them. And these are not surprising, and I'm guessing not unique to kidney cancer, but time constraints, lack of mentorship, limited infrastructure, lack of shared socialization and communication, lack of funding, of course, attitudes towards working with industry and access to new agents and trials. And again, I won't read to you, but you can see a number of strategies that will attempt to overcome these barriers. And this will be an iterative process, of course, and we'll, this is the first year of the academy, so I'm sure we'll do some things successfully and others not as much, and sort of try to learn from our mistakes and move on to really develop this over the next several years. These are the components as I see it. AKCI is Academy of Kidney Cancer Investigators. So committed career investigators and mentors, we absolutely have that. SMART goals, and really we tried to quantify goals for the mentees. And again, this will be sort of a work in progress, but you can see they range from presentations to publications to interaction and membership in societies, funding for trials and lab-based efforts, collaboration across the academy, which I think as this academy grows, and there will be new mentor-mentee pairs added every year, probably 3 or 4 such pairs added every year. Increased collaboration, and really creating a community that is geared towards the success of early RCC investigators. These are the principals. There's myself, Jim Brugarelis is the chair of my advisory board, and then you see the members. They are really representing different disciplines, different geography, some more basic sciences, some more clinicians, et cetera, but really sort of a broad spectrum of expertise, including the PIs of both RCC spores, Bob Uzo, who's a surgeon, et cetera. So really, I think a diverse and an excellent advisory committee, and I thank them for their service. Continuing on with essential components, structured guidance and feedback. So we're going to use a sort of an innovative software company that normally does mentorship for large corporations, but we're going to try and use it in this academy to really facilitate communication and feedback. And they also have some mentor training videos and coaches, and that's not really something in kidney cancer. I think really in any cancer where mentors get sort of training on how to be a good mentor, you sort of either do it or you don't, but this will try to provide some formal training for the mentors in this program. And then collaboration, as I mentioned, a big part of this, including collaboration with the Clinical Consortium Award that Eric runs and talked about. So this is what I see as really the iterative process of this academy. So establishing these goals, both short and long term, not just scientific project goals, but career goals, developing a plan and timeline, which we're in the process of doing, myself and my advisory board and the mentors providing the visibility and opportunity to achieve goals. We'll have ongoing meetings of various sorts to talk about progress, some didactics, et cetera, assessment of progress, and then really sort of starts over again with refinement of those goals moving forward. And this is the inaugural class that you're about to hear from. Lucas Salas, David Braun, and Abhishek Chakraborty. You can see the projects that they'll talk about briefly, and they each have a primary mentor and at least one secondary mentor that you see listed. So congratulations to all of them. And then the last thing I'll say is just that this grant mechanism will renew yearly, and so I'd really encourage young investigators out there who are interested in becoming part of this. I'm happy to answer questions now or offline. about, more about the academy, who should apply, how to do it, et cetera. But we'd really like to extend our reach. This year's class is more lab-based, but this is meant to also extend to clinical investigators as well. And again, I really see a lot of potential for growing this in the years to come. Thank you.
Thank you, Dr. Rini, for the introduction. We'll move on to the next presentation by the first Department of Defense mentee, Dr. Abhishek Chakrabarty. He is an assistant professor in molecular medicine at Cleveland Clinic's Lerner College of Medicine. He will be talking to us about exploiting epigenetic dysregulation to identify targetable vulnerabilities in clear cell RCC.
Thank you for the introduction, and I'm really excited and honored to be part of the first inaugural class of this award mechanism. So just a very brief overview in the interest of time to give you a sense of what we are going to be doing as part of this research project. I don't have any financial disclosures. So this project essentially emerged when I was still a postdoc in Bill Kaelin's lab, and we were doing some epigenetic profiling in VHL deficient HIF high CCRCCs versus the VHL proficient HIF low CCRCCs. And one of the platforms that we were using was a proteomic platform that allowed us to measure a number of epigenetic marks that were altered in these tumors. And what we noticed was that the CCRCCs essentially had a very distinct signature. They all clustered together, and this signature was being driven by a very specific chemical footprint on histones. So an increase in histone acetylation or hypomethylated state. And this is really exciting for somebody who's studying chromatin biology, because they would know that the acetylation mark is almost a flag that tells you that this is occurring at a location where genes are going to be switched on. It's a high expression mark. And so we made a simple hypothesis that maybe H3K27 acetylation in the VHL deficient cells was occurring upstream of very critical oncogenes. And we did this analysis by comparing VHL proficient and VHL deficient cells with the interest in asking where exactly this mark was occurring across the whole genome. So this is just an example. If you do this type of an analysis and you compare what's going on in terms of HIF targets, which we would expect to be robustly switched on a VHL deficient state, you can actually see very nicely the gray signal, which is occurring in the VHL deficient cells, is significantly higher than the red signal, which is in the VHL proficient cells. But this alone doesn't tell us anything about the biology. This is just a correlate, right? So gene expression going on as a result of H3K27 is not indicative of function. For doing that, we have to do some sort of functional assay, and we do two types of assays. We essentially can turn off the genes that are induced highly in the tumorigenic state, or alternatively, we can turn on or overexpress genes that are low expression in the VHL proficient or a non-tumorigenic state. And so in my lab, we do a lot of different types of chemical screens, which I began to do originally, in fact, while I was still in Bill Kaelin's lab. And so this is just an example of the screen. You could essentially infect cells that are proficient for VHL and now let them grow out over time, and then look for what are the genes that are essentially enriched in the tumors that form. So VHL deficient cells form tumors, proficient cells don't form tumors, and then you put back some of these genes and you can actually restore tumorigenic ability in VHL proficient cells. So again, very brief overview. We have some targets. This is working out. It is succeeding to some extent. One of our favorite targets right now is a gene named SLC1A1. It encodes it's an amino acid transporter that actually takes up aspartate and glutamate into cells, and we are scoring this as a potential oncogene. But more importantly, we can take VHL deficient cells, which typically form tumors, and in these cells we can inactivate this particular gene using CRISPR-Cas9, and whenever we are able to do that successfully, we can see a significant fitness defect in the RCC lines. So just summarizing, we can use epigenetic marking and experimental evidence in cells and in mice to essentially identify potential therapeutically relevant targets. One of them that we are right now focused on is a transporter, but there are many more to come. And so with that, I'll stop. These are my acknowledgments. My lab, my original work, which started in Bill Kaelin's lab, my colleagues from there, and then my mentors in this program, David McDermott and Nima Sharifi, and others who are involved, and of course, my funding. Thank you so much for this opportunity.
Thank you, Dr. Chakravarti. Moving on to our next presentation, we have our Department of Defense mentee, Dr. Lucas Salas, who is an assistant professor of epidemiology at the Dartmouth School of Medicine. He will be talking to us about cytosine modifications and renal cancer.
Well, thank you, Dr. Desai, for that kind introduction. So, as you mentioned, my lab is working mostly on what are these other epigenetic markers. In difference to what we saw in the previous presentation, I am working mostly on chemical modifications on the DNA, and in particular, what are these cytosine modifications and how can they alter in cancer. So what are these cytosine modifications? The cytosine modifications are chemical modifications at the DNA, specifically on the cytosine to guanosine context. This is a well-known repressor at the promoter level. And specifically when we talk about this modification of DNA methylation, we have several functions that we already know, such as the transcriptional repression of the promoter, in some cases, some transcriptional activation of the gene body, or some other things like protection to the genome, stability, chromosome inactivation, which is very important for imprinting in females. And one topic that I work a lot in my lab is the cell differentiation. So even though we have the same DNA in every one of our cells, these epigenetic marks are the ones that differentiate one cell to another. However, this is not the only one cytosine modification that is present in our body. We also have this other modification that is the DNA hydroxymethylation, which is less known. It could generate some pathways that are related to being an intermediate in active demethylation, but it could also be related to some stable marks with some unknown function in organs such as the brain. In some cases, it has been shown that it could also antagonize DNA methylation. It could help in DNA methylation fidelity, and it could help to promote transcription and pluripotency that could affect some development pathways. So what do we know of this cytosine modification so far? So in terms of this 5-methylcytosine, the 5-methylcytosine have been associated in the cancer genome atlas with differences in survival in clear cell renal cell carcinoma. Specifically, when they interrogated for all the different kidney cancer tumors, these 1,500 different CPG marks, they found that in the first cluster in this heat map on the right, you can see that there is a very first block that is marked by this blue ribbon on the top that includes several of these clear cell renal cell carcinomas in green and some of what they are called CPG island methylator phenotype or SIM. As you can see by the colors, there are differences between the normals that are on the left and these specific tumors in which we have a change with hypermethylation. Specifically, when we look at these CPG marks, there are several of these that are related to the Wnt pathways, such as the one on the left graph, that actually demarcate differences in survival for these patients. But as I mentioned on my first slide, there are other cytosine modifications. We know from some animal experiments that around 5% of the CPGs are methylated in different organs. This is something that is quite stable, and you can see that on the bar plot on the very right-hand side in yellow. However, for the 5-hydroxymethylcytosine, these levels are completely variable from organ to organ. They are much higher in organs such as the brain, and specifically in such organs that have a higher content of neurons. However, for the kidney that is here marked in red on the bar plots, you can see that this has some intermediate levels, and those are levels that are quite stable in animals. So why this could be important for renal cancer? In this case, we all know that we have these differences on the VHL pathways that could alter several mechanisms in renal cancer. But we also know that when we have these alterations in the substrate, and specifically what is demarcated by this large arrow, we can find some differences in the 10 to 11, the TET enzymes that could generate changes in the 5-hydroxymethylation and generate some chromatin alteration. So for this specific grant, what I want to investigate is there is some differential DNA methylation and hydroxymethylation patterns in the renal tumor bank at Dartmouth. I will take 200 tumor samples from clear cell renal cell carcinoma and 50 normal adjacent kidney tissues. The idea here is to measure using tandem oxy-DS conversion that would allow us to know what of those differences are related to methylation and which ones are related to hydroxymethylation. This is something that could not be teased on the TCGA previous analysis. The other part that is really important here is that I would try to determine if those relationships between the DNA hydroxymethylation in patients' tumors is affecting the survival. As I showed you before, there could be some specific alterations in the pathways that could be altering in a way that is not completely stochastic at the level of the tumors. Finally, the idea is to test different independent associations between these genetic marks and the gene expression of both messenger RNA and small RNA Sig. And using this sequencing, we can find if those alterations are happening in some specific organized ways. Finally, I want to acknowledge my mentors, Dr. Jim Riverolas, and my local mentor, Dr. Brock Christensen, and the people at the Kidney Cancer Academy. Thank you very much.
Thank you, Dr. Salas. Next, we have Dr. David Brown from Dana-Farber Cancer Institute, who will be talking to us about understanding CD8 positive T cell function and specificity in RCC.
Great, thank you so much to the organizers, to the DoD and Brian Renal as well for organizing such a wonderful program. So we know in advanced kidney cancer that immune checkpoint inhibition has been truly transformative for our patients, but unfortunately we're at a place where the majority of patients do not receive durable benefit from these agents alone. Now, in contrast to a typical paradigm of hot versus cold tumors, we know that the majority of RCCs are hot tumors, and so it doesn't fit the standard hot being responsive, cold being resistant paradigm. We have to sort of dig deeper. Further, we know that in contrast to most other solid tumor types, a high CD8 infiltration, the primary effector of anti-tumor immunity, has actually associated with a worse prognosis. And again, this stands really in contrast to every other tumor type and really highlights our inadequate understanding of T cell specificity and T cell function within this disease. Now, we know for T cell mediated immunity, this is an RCC, but in other tumor types, really the heart of that reaction, the heart of that immunity is the tumor cell, in this case, an RCC tumor cell, presenting antigens on MHC class I molecules, and those being recognized by a cognate T cell receptor, the T cells that are in the tumor. However, there's really a lack of understanding in RCC of what are the relevant tumor antigens that are being targeted, what are the cognate T cell receptors, and also the phenotype of those T cells that might recognize tumor? And then finally, how might this link to response or resistance to current therapies and also open up the avenue for new therapies? So in tackling these issues one at a time, the first question is really what is the landscape of antigens that RCC presents that can be recognized by the immune system? And here I'm hoping to take two approaches. One is a computational approach using prediction, and two is a direct detection method. Now, with respect to computational approaches, we've been fortunate to be able to integrate and develop toolkits to really do large-scale variant detection and measurement of things like ERV expression within these tumors to really get a sense of what are the aberrant states within RCC. And then importantly, using a neural network trained on mass spectrometry data, have developed a new toolkit for being able to predict which of those epitopes are likely to be bound by an HLA, which are likely essentially to be antigenic. This outperforms prior tools by a substantial margin, and something we can use to then try to infer computationally what is the landscape of antigens in RCC. But moving just beyond computational approaches, we hope to directly measure these as well. Using primary tumor samples and cell lines derived from RCC tumors from our patients, we isolate HLA class 1 molecules, elute off the peptides, and then use mass spectrometry to identify what are those HLA-bound peptides, what are those target antigens, and to do so much more systematically. The second half of that equation is then identifying what are the cognate T cell receptors and what is the state, the phenotype of those T cells in the tumor that have a chance to mediate anti-tumor efficacy. Now, we know from our single-cell RNA-seq endeavors that, one, we're able to effectively sequence individual T-cell receptors, the alpha and beta chain, to really get a comprehensive look at what the T-cell receptor repertoire looks like in RCC. And even in these initial studies, what we've seen is a tremendous expansion of individual clonotypes, where a single T-cell receptor is found on 20, 30, 40% of T-cells in an individual tumor, which begs the question, what are those T cells recognizing? What are their target? And that's really going to be a key question. So we've developed a number of approaches to really tackle this, but one uses peripheral blood T cells from a patient or their TILs, their tumor-infiltrating lymphocytes, and actually co-culturing it together with their autologous tumor cell. We're then able to identify tumor-reactive or antigen-reactive T cells, isolate them through flow cytometry, and then perform single-cell RNA and TCR sequencing. And this is an example of exactly that process for one of our patients, where we're able to isolate T cells, both CD4 and CD8, that are tumor reactive. And we look at the most predominant clonotypes. In this case, we're really able to see these dark blue dots, that these are clonotypes that are predominantly CD8 clonotypes. And so this is a way to really identify exactly what are the tumor reactive T cell receptors. But that's not enough just to know what they are. We really want to link them with the cognate antigens that we discover in the first part of this project. And we have a platform to be able to do that. Taking those T cell receptor sequences that we've generated, we can introduce them into a lentiviral vector, transduce them into healthy donor T cells, and actually probe them against our candidate panel of antigens and see what is their specificity. And we've been able to successfully do this for a number of target antigens with RCC using a flow cytometry-based readout. And then finally, of course, once we have a knowledge of the landscape of target antigens and their cognate T cells, the question is, how does this effectively link to response or resistance to current therapies? And the ultimate hope that we have is to identify these sides of the equation, the RCC tumor antigens, their cognate T cells, link it to response and resistance to immune checkpoint inhibition, but ultimately also use this as a platform for developing novel antigen-specific therapies that we can use to help our patients. With that in mind, there's a lot of people to thank, but mostly my mentors, Catherine Wu and Tony Shaweri, our funders, including the DoD, and of course the patients and the family for their generosity. Thank you.
Thank you so much to all the speakers. I think now we move on to the next part of the session. I'd like to introduce Dr. Brian Rainey and Eric Jonash for moderating the rapid fire top abstract discussion.
Great. Thank you very much. And I think we have the four presenters on. And so the way this is going to work is Brian and I are going to have each of the individuals here present for 60 seconds. We're not going to be using slides, and then we're going to do question and answers, so to really talk mainly about the implications and the significance of these findings. And the order we're going to go in is essentially as per the outline. We're going to start with Alex, followed by Luis, followed by Monty, and wrap up with Yui. So let's start with Alex. Alex, why don't you just give us a 60-second summary of your of your abstract, which, by the way, is available on the platform. There's a really great video for each one of these and also slides you can look at. So Alex, go ahead.
Sure. Thanks, Dr. Jonash. So in our study, we sought to evaluate if enrichment in certain gut microbial species corresponded to peripheral plasma cytokine levels in metastatic RCC. All participants were planned for treatment with either a VEGF TKI or immune checkpoint inhibitor. Prior to the initiation of systemic therapy, measured stool microbial composition using whole metagenomic sequencing analysis. We then compared variations in stool microbial composition amongst patients with high or low levels of the following cytokines: interferon alpha, interferon gamma, TNF alpha, IL-2, IL-6, and IL-8. Levels of Akkermansia species were significantly higher in patients with low levels of IL-6 or IL-8. Elevated interferon gamma was associated with the enteric pathogen Shigella sonnei. Using Shannon Diversity Index, we did not find any differences in microbial diversity across all 6 cytokines. These data suggest that gut microbial composition plays a pivotal role in systemic immunomodulation via circulating cytokines.
Thanks, Alex. So one of the key questions here is what's the directionality of effect? So you've seen a relationship between the presence or absence of various cytokines and the presence and absence of various gut microbial elements. And who's actually driving whom here?
Yeah, that's a really important question, Dr. Jonash. I think, as mentioned during session 3 yesterday, several studies have found peripheral cytokine concentrations of IL-6 and IL-8 are negatively associated with benefit in RCC. Additionally, work from Dr. Bertrand Rowdy and Dr. Lisa DeRosa have identified Akkermansia species is associated with favorable responses to ICIs. So, cause and direct is still very preliminary here, but it seems there may be some sort of pathway that Akkermansia species are, when they're enriched, are inducing low concentrations or contributing to the low concentrations of IL-6 and IL-8, and maybe a potential link between the gut in response to systemic anti-cancer therapy.
Go ahead, Brian.
I was just going to say, first of all, congrats. This is great work. I know it's not easy to do. Can you tell us what you think about, where to go from here? I think these studies are interesting, sort of hypothesis generating. Like Eric said, I'm not quite sure the directionality, but I think there's a number of these studies across malignancies that would suggest there's something there. But how do we take the next step and turn this into a therapeutic benefit, if you will?
Yeah, also a great question. I think first and foremost, the most urgent need is for a practical diagnostic tool that can characterize patient stool microbial profiles. There's several efforts underway, such as the Oncobiome project that's being run out of Europe, that are working towards developing effective and practical solutions. But without that in hand, I think we're not going to be able to really make all that much of a practical change in treatment. And then another aspect I think that's intriguing is there's several trials looking at fecal microbial fecal transplantation. So there's a couple studies, one by Dr. Vriara Madsen and another by Dr. Bertrand Rowdy in Science in 2018 that showed that patient responsiveness to checkpoint inhibitors can be conferred onto mice via transplantation of the fecal FMT, it's called. So it would be interesting to see if similar benefits exist between human responders to non-responders.
So with regards to your own particular work, what are your next steps in this?
Yeah, we're in the process of trying to see if we can employ Akkermansia supplementation in our patients in a prospective fashion. So it's still very early on in that process, but that's our group's next step, is to see if that might confer any sort of benefit in our CC patients at City of Hope.
All right. And one last quick question, just as sort of a nerdy statistical question. You used LDA, you used linear discriminant analysis for your analyses. Why that and not other statistical tools?
I think it was in large part just because when you're dealing with a lot of this metagenomic sequencing, there's just so many different species that we're trying to differentiate between. And then the left-C analysis really allows us to differentiate those species that stand out dramatically compared to those that are not as much. So I think that it was sort of a similar approach employed by the previous studies that I already mentioned, and we're sort of piggybacking off their statistical methods, but it kind of does allow sort of see through a lot of the noise that comes through with this metagenomic sequencing.
Great. Well, let's move on. Thank you so much for your presentation and for your excellent answers to our questions. And let's move on to Luis. Luis, why don't you give us your 60-second summary?
Sure, thank you. So as we know, BAP1 mutations have been associated with higher formant rate and poor clinical outcome in patients with clear cell RCC. In our study, we explored radiogenomics as a non-invasive method to identify these mutations. For this, we used CT imaging of 88 patients from our center, 9 of which were not BAP1 mutated. Segmented images from both primary and metastatic sites were then processed by PyRadiomics, which is a Python-based package used to extract radiomic features. And these radiomic features were then correlated to the BAP1 mutational status of our patients. Out of 1,000 features analyzed, we found that 2 features stood out. These were degree level co-occurrence matrix and degree level dependence matrix. This wasn't the analysis of the metastatic sites. Our data suggests that using radiogenomics might be a viable option to identify not only BAP1, but perhaps other mutations and drive the future in precision oncology.
Great, thanks for that succinct summary. So, you know, in terms of the mutations that we see in renal cell carcinoma, obviously PBRM1 mutations are the ones that we see most commonly. And, you know, there was a paper that Brian just published yesterday, and a number of us have published over the past few years, sort of looking at the fact that this actually, PBRM1 mutations influence the angiogenesis phenotype of renal cell carcinoma. And that when you were performing this analysis, did you look at this in an unbiased way and look at the other potential driver mutations like 52 PBRM1? And did you or did you not find a signature for those?
Sure. So we actually use the whole exome sequencing that we use, not for every patient, but for some patients. And we do have some data from other mutations. However, in our study, we only found relevant data from 2 mutations, one of which is currently under analysis, and the other one was BAP1. But we did include different type of mutations.
So, Luis, this is Brian. Great work. Relevant to what you just said, is there a reason that radiogenomics would be more apt to one kind of mutation versus another? Is that...
The thing is that, as we know, there's a relative paucity of relevant mutations in RCC. And the radiogenomic features that are extracted depends to some extent in the program or the package we use, right? And the phenotype that we see on imaging using the computerized extraction is not something that can be seen just by the naked eye, but it's a comparison between pairs of pixels and BOC12s, which are like 3-dimensional imaging units, which are a little abstract to comprehend just on a regular basis. So in that aspect, I would say that although we can see, and there has been some previous published studies using like readily observable features such as intratumoral calcification or ill-defined borders to identify these kind of mutations, this may not apply certainly to a computerized extraction.
So is there a way of potentially training this? Because, you know, you can use an unbiased approach where you have this whole bunch of features and then you can determine whether or not this, I don't know what you want to call it, palette of features is associated with your genome. But if you then have some sort of a hypothesis as to how you're going to be influencing the characteristics, can you go the opposite way? Train your discriminators and then determine whether or not you're capable of detecting mutation A versus mutation B?
Sure. So we're currently working on an AI method. So we're going to train computers using already CT images that we do have so that the AI program can actually identify the features that he needs to look for specific mutations. But nothing as of right now, at least as far as my knowledge goes, has been published using AI in this particular manner.
And Luis, I have a question. So, in RCC, my opinion is that I think if we're going to develop therapeutic biomarkers, they're probably going to be RNA-based and not DNA-based. Could this technology be applied, if you will, to look for certain RNA signatures like the many that have been published? Does that make sense or no?
That's a good question. However, that's one I'm not fully able to respond as of right now with the data that I have.
Okay.
Great. Thank you. Once again, excellent presentation, great answers. And up next is Monty. Monty, give us your 60-second summary.
Yeah, great. Thanks a lot. So a lot of information to digest, and apologies for my technical issues there. I think my fellows are a lot savvier with this platform than I am. Great job, guys. But this is what I think is a very clinically relevant study that juxtaposes 2 doses of lenvatinib and everolimus. I think all of us here on the call have, some experience using lenvatinib and everolimus in the clinics. We know that the standard dose is 18 milligrams of lenvatinib with 5 milligrams of everolimus, and we know it's a tough dose for many patients. So the objective of this study was to see whether or not we could get away with using a slightly lower dose of 14 milligrams in combination with 5 milligrams. So Eric, this was a non-inferiority study, and non-inferiority studies always employ very complex designs, and ours was no different. We used a primary efficacy endpoint of response rate at 24 weeks, and we also had safety endpoints based on treatment emergent adverse events. And just to cut to the chase, in this study, juxtaposing 18 and 14 milligrams, we found that the 14 milligram dose was not non-inferior based on the pre-established guidelines. So just, highlighting the response data in this cohort of over 300 patients, the response rate at 6 weeks to 18 milligrams was 35% versus 32% at 14. Now, you may kind of scratch your head and say, well, that sounds awfully similar. But if you do a deep dive of the data that we have in the poster and in the presentation, I think you'll see some more compelling trends here. So, overall response rate was 40% with 18 milligrams, 35% with 14 milligrams. And I do think you see somewhat of a striking difference when it comes to progression-free survival. I urge folks to focus on those curves. 14.7 months of PFS with 18 milligrams versus 11.1 months at 14. In terms of treatment emergent adverse events, really no difference between the two. If I were to summarize this in one bullet point, I'd say, really just no need to switch our current strategy of lenvatinib at 18 milligrams. Eric?
Great. Monty, thank you so much, and congratulations on getting this this study done. So the first question that I really have is sort of looking at some of the data here, the frequency of dose reductions, and then the dose intensity of lenvatinib and the 14 milligram and the 18 milligram dose. So it looks like your dose intensity at the median dose intensity at the 14 milligram is 13.33, and at 18 it's 13.83. Yet the frequency of dose reductions ends up being about the same. So help us reconcile those data.
Yeah, so bear in mind, Eric, that this study employed a complicated design in which patients started at 18 milligrams and 14 milligrams, but if they didn't incur any treatment emergent adverse events, they were actually able to escalate to the 18 milligram dose after a 3-month stretch of time. So I would propose that although you do see some, equivalency in median dose and so forth, it's really hard to balance those estimates because of the fact that we allow for this intra-study dose escalation.
So, hey, Monty, it's Brian. Just let me push you on that a little bit. So when I read this and watched your presentation, as Eric said, it seemed like all the numbers were the same. The dose reduction, the toxicity, boy, they looked really close to me, within a few percentage points. So was it that the 18s had to come down a little bit and the 14s got bumped up, and so the median dose is the same, presumably the exposure is the same? And if that's the case, then maybe the efficacy differences that you noted aren't really different. They're just due to a relatively small study in this context. Do you know what I'm asking?
Yeah, you know, I get where you're going with this, Brian. Yeah, absolutely. And I will say that the majority of patients were able to sort of bump up that dose to 18 milligrams over the course of time. You know, I think that, you know, much as we use axitinib, for instance, and employ that dose titration, you know, several cycles into therapy, I think here that, you know, the overarching data argues to the fact that that sort of strategy with lenvatinib probably isn't relevant. You know, again, that primary endpoint with objective response rate at 24 weeks is a complicated one. You know, I think many of us would scratch our heads and say, how relevant is that? We picked it because, of course, we're employing this dose titration a couple of months into therapy, the up titration specifically. But I would say that, you know, again, the PFS difference that you're seeing here of 3 months, the overall survival curves, which seem to be favoring lenvatinib at 18 milligrams for the most part, argue that I think we're really at the right dose.
Fair enough.
The conclusion is go big.
Go big. Go big or go home. Thanks, Eric. Yeah.
Great. Thank you so much, Monty, for those excellent answers. And last is Yue. I may be mispronouncing your name. I apologize in advance. And please give us your 60-second summary.
Yeah, thank you, Eric. That's pronunciation is perfect. And I also sincerely thank the International Kidney Cancer Association for this opportunity to present the results of our recent study. So our study uses multi-regional sequencing data to study the intratumoral heterogeneity and tumor evolution, and uses positional information to find evidence of spatial evolution of clear cell renal cell carcinoma. With the extraction of spatial information from the macroscopic photos taken after surgery, we were able to reconstruct digital tumor maps based on the X and Y coordinates of both the biopsy regions and the boundary between tumor tissue and normal tissue. We found that central part of the tumor tended to behave more malignantly by having a higher proliferation rate, a higher necrosis rate, a higher somatic copy number alterations, and more ferment grade 4 regions and more metastasizing clones. Moreover, a pairwise spatial distances between each 2 biopsy regions were found to be positively correlated with the genomic distances, meaning that the further 2 biopsy regions are apart, the more different their genomic backgrounds are. And finally, we observed 2 patterns of clonal expansion, namely contiguous expansion and clonal dispersal based on the phylogenetic trees generated and the spatial locations of the biopsy regions, which we think might shed light on the spatial evolution of tumors. And we hope our study provides a unique insight into the field of intratumoral heterogeneity and tumor evolution. Thank you.
Thank you so much for that great summary. And you know, this is a really, really cool set of data. And the first question to you is, what does this tell you about how a tumor grows and evolves? So are we seeing tumors expanding centripetally or centrifugally?
Well, that's a really good question. Actually, from the experimental data, we can't really tell the cause and effect relationship of whether it grows centrifugally or centrically. But we are trying to do some computerized modeling to test to mimic what we see in the experimental data, and also we are using that computational model to test some of the ideas that cannot be answered using the experimental data. So we also want to answer that kind of question. So right now we can only make a guess. So maybe we think that the tumor starts at the center and it expands outside.
The fact that you have greater copy number abnormality and greater aggressivity in the center of the tumor, that would suggest that evolution, that there's 2 possibilities. One is that you're actually having the more recent clonal evolution occurring centrally, or it's the microenvironment itself that's actually driving its behavior. So which of the 2 do you think is going on?
Yes, exactly. I think both of them are going on actually. So in this study, we lack enough number of RNA-seq data right now. So in the future, we want to add the RNA-seq data in and to see the microenvironmental changes or comparisons between tumor center and tumor margin to see whether the environmental factors also is driving the big difference.
And this is Brian, kind of a similar question. I was wondering if the necrosis in the center of many large primary tumors is sort of driving some of this. And related question is, I think these are all primary tumors, or have you analyzed any metastatic deposits?
For the metastasizing clone analysis, we used, some of the cases in our cohort has metastasizing sites sequenced. So in the analysis of metastasizing clones, we included both the primary sites and the matching metastasizing sites to see, and we saw that the metastasizing clones tend to be originally located in the tumor center.
Interesting. Thank you.
Thank you.
So I think we're out of time. We're right at 12:30. I want to thank and congratulate all of our participants. Thanks for excellent summaries and for staying on time. And thanks to the KCA for sponsoring the session.